So you know that there are different levels of organization in human body, right? From its smallest to largest. So smallest is the chemical level that we have taught in last class. One step higher of chemical level is the cellular level. So today first we'll talk about the cells. Then we'll talk about the tissue, which is another step higher than the cellular level. So first you will see the definition of a cell and a part and the parts of a typical cell. What is that? Okay. One important part of a cell is the cell membrane that gives the boundary of the cell. So we'll see the structure and functions of the cell membrane. In the cell membrane, you have two types of proteins, integral proteins and peripheral proteins. We'll see those proteins in the cell membrane and their functions. Okay. Then we'll talk about the cell junctions, how one cell is attached to another. Cell junctions are the structures that help to attach the adjacent cells together. So cell to cell connection or attachment is given by the cell junctions. So we'll see the important cell junctions that attach the cells of our body. Then we'll talk about the transport across the cell membrane. Cell membrane is a semi-permeable membrane. That means it selectively allows, it doesn't allow everything, but allows something selectively permeable or semi-permeable. So we'll see how the chemicals pass through the cell membrane. That is the transport across the cell membrane. Then we'll see the response of a living cell in three different types of solution. If you put a living cell in isotonic, hypotonic and hypertonic solutions, how that cell will respond in those three types of environment. We'll see that. Okay. First, the definition of a cell. The cell is the smallest structural and functional living unit. So, smallest structural and functional unit of the body is the cell. Human body consists of 50 to 100 trillion cells, not million, not billion, but trillion. So just think that it's a huge number of cells. Over 200 different types of cells are present in the human body. How they are different? They are different in size, shape, function, and other ways. But one thing is common, all cells are composed of chiefly C, H, N, and O, carbon, hydrogen, nitrogen, and oxygen. You must remember from last class I said that 96% body mass is formed by those four chemicals, right? So cells are mostly formed by those four elements. Here you see just an example how the cells could be different from each other. You know, there are more than 200 different types of cells, how they could be different from each other. You see the top left, those two cells are fibroblasts, connective tissue cells, and they produce fibers, connective tissue fibers. Next one, two cells, you see round, red colored, those are red blood cells or erythrocytes. They are round but flat, round shaped, red colored cells are red blood cells. So they are different in color, shape and size. Then next one, below those two, you see cubical shaped cells, 5 cubical shaped cells. Those are epithelial cells, cuboidal epithelial cells, one type of epithelial cells. Then you see two different types of muscle cells. Left one, which is big, long, that is a skeletal muscle cell. It has multiple nuclei. If you look carefully inside, you see the purple colored multiple nuclei, and it is pretty big, very big. In the right hand side, you see several smooth muscle cells. They are smaller, spindle shaped, and only one nucleus in the center. So even you see the muscle cells are different from each other. Different muscle cells are different from each other. Then go to the bottom left, you see that yellow colored fat cell. In fat cell, the nucleus is not in the center, attached to the membrane. You see the nucleus attached to the membrane. Next one has multiple processes, extensions. You see that is a macrophage. This cell has phagocytic property, phagocytosis. It can engulf the microorganisms or other cells. That's why it has multiple processes to help that process. Okay, phagocytosis. In the right side, the top one is a nerve cell. You see, a nerve cell has a cell body. Inside the cell body, there is a nucleus and then multiple processes. And those processes have branches. And function is completely different. The function is transmission of electrical signal, electricity. The last one you see is a sperm, male sex cell. Sperm has a head, body or midpiece and a tail. It looks like a baby fish and it can swim in the semen or fluid. So you see how different cells could be different in shape, size, color and function. Parts of a typical cell. If you see a typical cell, it has a membrane called the plasma membrane that gives the boundary, outer boundary, and plasma membrane separates inside the cell from outside. So if I ask you what are the functions of the plasma membrane or cell membrane, it provides boundary of the outer boundary of the cell. Number one, separates inside the cell from the outside, okay, and maintains internal environment of the cell. And also you can write down that cell membrane or plasma membrane is a semi-permeable membrane. It selectively allows some chemicals to pass through it. Then you have cytoplasm. Cytoplasm is the intracellular fluid, that means fluid inside the cell, and it contains organelles. Two types of organelles are present in the cytoplasm or in that fluid: membranous and non-membranous organelles. Okay, we will see important membranous and non-membranous organelles. Then usually in the center of the cell you have a nucleus. Nucleus is the control center of the cell. Why it is called the control center? Because it controls the cellular functions. How that cell will behave and function, that is controlled by the nucleus. Make sense? How that cell will behave and function. Inside the nucleus you have the chromosomes, and inside the chromosomes you have DNA, and DNA is the most important part of the cell that controls the function and behavior. DNA encodes the information how that cell will work and behave. Genetic information is coded inside the DNA. Okay, now if you see this picture, it is a nice, beautiful picture, very colorful picture. Although if you see a cell under the microscope, it will not look that beautiful. Okay, so in the textbook we can draw nice pictures, but real things are not that beautiful, but you can get a very good idea from this picture. You see the cell membrane or plasma membrane, then inside the cell you have the cytoplasm here, and in the cytoplasm you have organelles, different types of organelles. Some are membranous, some are non-membranous, and then we have the nucleus. This is the nucleus. Okay. We will just briefly go over this part, because I believe in your high school or in intro bio you have learned about the cell more. So in anatomy and physiology we will just quickly go over it. Important membranous organelles are mitochondria, endoplasmic reticulum, Golgi apparatus. Mitochondria is called the powerhouse. It is called the powerhouse of the cell because most of the ATPs, ATP gives energy. So most of the ATPs are produced in the mitochondria. So those cells of your body work more. They have more mitochondria because they need more energy. Make sense? Like your heart muscle cells, cardiac muscle cells are working 24 hours, right? Even when you are sleeping, the heart is not stopped its beating. So if you see the cells of the cardiac muscle or heart muscle, you'll see many mitochondria there to produce more ATP. Then you see the blue structure around the nucleus, that is the endoplasmic reticulum or ER. Now, endoplasmic reticulum has two parts: rough endoplasmic reticulum and smooth endoplasmic reticulum. Why one part is called the rough ear and another is smooth? Because on the surface of rough ear you will see
the ribosomes are attached. That's why the surface is rough. We say rough ear or rough surface ear. Both are correct. Okay? Rough surfaced endoplasmic reticulum because of the presence of ribosomes. Okay? And so this part is rough ear. You see many tiny dots, red colored. These are ribosomes attached on the surface. So this is rough ear, and this part is smooth ear because no ribosomes are attached. Okay? Ribosomes manufacture proteins. Ribosomes manufacture proteins. And then the proteins enter into the ear, rough ear. Okay, so rough ear does folding of the proteins. So proteins, proteins are long chain amino acid. I have talked about that in last class, I believe. So amino acids form chain, and that is the protein molecule. Okay. So after the protein is formed or produced, it enters into the ear, and folding of the proteins are done by the rough ear. Okay. Then the proteins are sent to another organelle, that is the Golgi apparatus. You see the green organelle or structure, that is the Golgi apparatus. And inside the Golgi apparatus, the proteins are packaged. So packaging of the protein is the function of Golgi apparatus. Okay, so ribosomes produce or manufacture, rough ear folds the proteins and send to the Golgi apparatus, right? And Golgi apparatus does the packaging of the proteins. So it is like a factory. If you go to a factory of something, one section manufactures, right? And another section does what? Folds the things, and another section does the packaging, okay, to send it out. Okay, so now if you see the Golgi apparatus looks like the number of flattened sacs piled one over another. So, and both ends of those flattened sacs are round, kind of round, and the proteins or protein molecules accumulate at the both ends. After packaging, they're sent to the ends, and then eventually the ends get detached from the main body and form vesicle. So that's how the vesicles are formed from both ends of the Golgi apparatus. Now the vesicles, we can say that vesicles are round membranous sacs, right, that contains proteins. And this protein could be a hormone or any other kind of protein could be there. It depends what kind of protein is being produced by the ribosome. Ribosome can produce many different types of proteins. And then you see the vesicles move towards the cell membrane or plasma membrane, okay, and releases the
protein molecules to the outside of the cell, and that process is called exocytosis. Gets attached to the cell membrane and burst or rupture, and release the chemicals to the outside, protein to the outside, and that process of releasing to the outside of the cell is called exocytosis. Okay? Now, smooth part of ER has different functions. Smooth ER helps in lipid synthesis, detoxification, okay. What is detoxification? You know that metabolism occurs inside the cell, right? In our body, inside the cell, and when metabolism occurs, some harmful toxic chemicals are produced, right? So those chemicals are detoxified, neutralized in smooth air. Make sense? Also, you take food and other chemicals, right? Every day you take from outside, like medicine. All foods and chemicals or medicines have some toxic components. A small amount, but toxic components, and those should be detoxified, and a smooth ER performs that function. Okay. Now we will see the nucleus. If you see the structure of the nucleus, the outer covering of the nucleus is called the nuclear envelope, and then inside the nucleus you have chromatin and nucleolus. Nucleolus is a round structure in the center of the nucleus where the chromosomes are heavily, densely packed. Okay, there. So those are the parts of the nucleus, and in the chromosomes you have the DNA, and that codes the genetic information, how that cell will behave and function. Cell membrane is an important part of the cell, and if you see the structure of the cell membrane, it is a phospholipid bilayer. Cell membrane is a phospholipid bilayer, that means two layers of phospholipid in which proteins are embedded or inserted. So I am repeating again, the definition of the structure of the cell membrane is, it is a phospholipid bilayer in which the protein structures are embedded or inserted. Now here you see, this is a cell membrane, and these are phospholipid molecules, phospholipid units or molecules. They are like this. These are phospholipid molecules or phospholipid units, and they form how many layers? Two. That's why we say bilayer. Okay. So like this. This is one layer of phospholipid. This is another layer of phospholipid, and in which proteins are embedded. So this is a protein structure. Then again phospholipid. Okay. This. This is another protein. This is another protein. Again phospholipid. Okay. So phospholipid bilayer, in which proteins are inserted or embedded. That is the structure of the cell membrane. Now, if you see a phospholipid unit or molecule, it has a head part and a tail part. Okay, head part and tail part. Head part is also called polar end, and tail part is non-polar end. Okay. Polar end and non-polar end. Head part loves water. Tail part dislikes water. So, head part loves or likes water, and tail part dislikes or hates water. That's why head part is also called hydrophilic. Hydro means water and philia means attraction or liking. Hydrophilic. Tail part is hydrophobic, dislikes water. Okay? So this part, hydro means water, hydrophobic. Part. So water molecules cannot easily pass through the phospholipid bilayer, because you know that these tail parts are hydrophobic, doesn't like water. Okay, so will not let water come close to it, stay away. Okay, so that's why the water molecules cannot easily pass through the phospholipid bilayer. Now proteins. Two types of proteins are present in the cell membrane. Some proteins are deeply inserted. You see this one going all through, and this one is also deeply inserted, although it's not going all through, passing all through the cell membrane, phospholipid bilayer, but it is deeply also inserted. So these are integral proteins. Integral proteins. And some proteins are loosely attached to the integral proteins. Those are called peripheral proteins. Peripheral proteins. Okay, so two types of proteins, integral and peripheral proteins. So that is the main structure of the cell membrane. Now there are some other structures present in the cell membrane. Those are not the main structures. You see here the green chains. These are glucose. These are glucose, and these yellow chains you see here, structures, these are lipid or cholesterol. Lipid or cholesterol. So glucose and lipids are also present in the cell membrane. Now, in some places you see the glucose and lipid are attached to each other. For example, here you see lipid and glucose, they are attached. That's why this whole structure is called glycolipid. Makes sense. Glucose, glyco comes from glucose, and lipid is the fat. Okay, together form glycolipids. So that is the structure of the cell membrane. Now, we will see the function of those proteins in the cell membrane. Integral proteins perform some important functions. Transport. You remember I said that water molecule cannot pass through phospholipid bilayer because of hydrophobic part here. So what happens? Some integral proteins have channels like this through passes, and through those channels
water and water soluble molecules can get in or out. Okay, so you see integral proteins help in transportation of the chemicals in and out. Some integral proteins work as receptors. This is very important. How the integral proteins work as receptors. Some integral proteins have let me draw another integral protein. This one is helping in transport. This integral protein, you see, has binding site. For example, this one has binding site like this. There is another integral protein here. That one has binding site like this. Another integral protein is here. That has binding site like this. Okay. So integral proteins, those work as receptors. They have got binding sites for the signal molecules. Like you take medicine, or a hormone will work on this cell. So hormone first must bind with those receptor proteins. Make sense? Hormones or chemicals, those are signal molecules, will bind there. Now the question is, you know that all, just think about medicine. You take different types of medicines, right? All medicines don't work same way or on same organs, right? Some medicine will work on liver, some will work on kidneys, right? Some will work on the brain. How that happens? So, for example, you have taken a medicine and the molecules are like this, round. It's a signal molecules, and will come to the cell membrane and will try to find where it can bind. And binding sites are present on the integral proteins, different shaped binding sites. So this one will fit where? Here, because this is round. Make sense? If it goes here, no binding. If it goes here, no binding. If it goes here, then nicely it will bind with the receptor, right? Integral protein binding site. And when that binding will be perfect fit, you are putting the key in the lock, right key in the lock. Make sense? And that will send a signal to inside the cell. This is inside the cell, and this is outside because the chemicals are come from outside of that cell. Okay. So here you have the cytoplasm. So signal will go to inside the cell, and intercellular machineries will start to work. Okay. So that's how the integral proteins work as receptors. If you take the medicine or chemical or hormone that has molecular shape like this will fit here. The signal will get it. Okay. So that's how some integral proteins work as receptors. They have the binding sites. Some integral proteins so we'll see how integral proteins help in transportation. Here you see two integral proteins, and they are helping in transportation of the molecules, but differently. So first one you see has channel inside it, and that channel is always open, like that, always open. So the molecules, those are smaller than the size of this channel, will get in or out easily because this channel is always open. So that is one type of integral protein and how it helps in transport. Next one, you see another integral protein. That channel is
not always open. It remains closed. To open that, you need what? ATP, energy. So this one usually remains closed, but ATP is needed to open that door. Okay, so you see, integral proteins help in transport, but different integral protein functions differently. This is the integral proteins, how they work as receptors. I have explained that here, so you already know their binding site. Some integral proteins have enzymatic activity. They work like enzymes. You know that enzymes convert one molecule to another. So here, very simple example, they have shown two different integral proteins are working as two different enzymes. So this one is changing this molecule to this, and this one is changing this molecule to that. So, so those are the functions, important functions of the integral proteins. Okay. Make sense? Okay. So now we will see the functions of the peripheral proteins. These proteins loosely attached to the integral protein. Peripheral proteins perform two very important functions. One is they form intercellular junctions or joinings. Intercellular junctions or joinings. I had mentioned already that cells are attached to each other in your body by intercellular junctions. So what happens, this is, if you see the epithelial cells, you have already seen when I showed you different types of cells, cuboidal epithelial cells, right? Nucleus. So you see these cells are attached to each other side by side. How they stay attached to each other? Because they have intercellular junctions or joinings, and those intercellular junctions or joinings hold the cell membranes together, attach the cell membrane together, and those are formed by peripheral proteins. Okay. Another important function of the peripheral proteins is cell-to-cell recognition. It is very interesting and important. One cell can recognize another cell. It's like you can recognize different persons, right? So your body cells behave like that. They can recognize other cells, and that is helped by the peripheral proteins of the cell membrane. Okay. Now let me give you an example how you know that your cells can recognize other cells. If you take a tissue, for example, skin grafting, you are taking tissue from one part of your body and put in another part of your own body, the chance of acceptance is very high. Usually it will accept, right? Because your cells can identify the other cells. Now if you take skin from another person's body, the chance of rejection is very high, right? You know that, will not accept that. So that is a very good clear example that your body cells can identify the nearby cells or adjacent cells. Okay, and that is a big problem, that transplantation, when we transplant one organ to another person, right, or tissue from one person to another person, rejection occurs, and we need to take care of that. And that is given by the peripheral proteins. So remember the important functions of integral proteins and peripheral proteins, and if I ask, just mention those. Okay. Now, junctions. We know that peripheral proteins form intercellular junctions or joinings. What are the important junctions we have in our body? Three types of junctions are very common in between the cells of our body. One is desmosome, another is tight junction, another is gap junction. Okay, so tight junctions, desmosomes, and gap junctions. Those are common. Tight junctions prevent fluids and most molecules from moving between the cells. So if you have tight junctions here, they will not let the fluid or molecules go from one cell to another cell. That's why they are called tight junctions. Very small few molecules will be able to pass. So most molecules will be prevented. Now, if you have gap junctions here, you see, if this is a gap junction, in gap junction, there are many pores or poles. So we let the fluid or molecules easily move from one cell to another. So gap junctions have many pores. Now, desmosomes. If you see the desmosomes are the strongest junctions. They hold the cell membranes very strongly together. How? Have you seen zipper in your cloth? You know, we use zipper. So desmosomes are like this. They have linker glycoproteins from both sides and get attached like this. Hold the cell membranes like this. So very strong attachment is formed by desmosomes. So if you see the desmosome, in this side of the cell membrane you have a disc. In this side of the cell membrane you have another disc. And from the disc, linker glycoproteins arise like this. And they interlock. Okay, linker glycoproteins arise from the discs from both sides and get attached like this. So very strong attachment. You cannot easily separate the cells because of desmosome. Now the question is, where, in which areas you have more desmosomes? We know that in skin cells. Skin cells are attached to each other by desmosomes. How you know? You have seen, if your skin gets dry, sometimes you can peel the skin, right? You can peel the skin. That means the cells are attached to each other strongly, so they will come, all cells will come together. Okay? Snake, you know, snake, when snake, you know, how to say that? Sheds, yeah, skin, I will say a chunk of skin comes out. So another location is cardiac muscle. Cardiac muscle fibers or cells are very strongly attached to each other by desmosomes, and that helps the heart to contract quickly. All cells are strongly attached to each other, so they will work at the same time. Make sense? They can work at the same time together. So just to know, those are the important intercellular junctions or joints and how they work. Here you see the tight junction. You see the bottom picture. Two cell membranes are attached to each other by tight junctions, and it's like stitching two pieces of clothes. okay, together, stitching two pieces of cloth. This is a desmosome. See the lower picture. From two discs, those are called plaques. Discs are also called plaque. From the disc, linker glycoproteins, also called cadherins. Underline these two, plaque and cadherin. Linker glycoproteins are also called cadherins, and they arise from the discs or plaque and get attached to each other like zipper. Here you see the gap junction, and you see channels are present in gap junction or pores, so fluid can move from one cell to another. Okay, now we'll see the transport across the cell membrane. Cell membrane is a semi-permeable membrane. That means it selectively allows the chemicals to pass through it. Will not let everything to pass through it. The chemicals pass through the cell membrane in two ways: passive transport or process and active transport or active process. In passive transport, no ATP or energy is required, but in active transport, it is called active, right? So it needs ATP or energy. That is one main difference. Another difference is passive transport occurs in living and non-living membranes. You can make a membrane, artificial membrane, and in the lab you can put two different solutions in both sides, and you will see the passive transport occurs through the cell membrane, non-living cell membrane. But active transport only occurs through the living cell membrane because ATP is required, right? Only living body can produce ATP by metabolism. Metabolism occurs only in living cells or living bodies. Since ATP is required in active, so it can occur only in living cell membranes. Now, both passive and active has subtypes. Common passive processes are simple diffusion, facilitated diffusion, and osmosis. So those are different types of passive transport or passive process. Simple diffusion. What is simple diffusion? If this is a semi-permeable membrane, okay, and you have glucose solution here, is a glucose. So you have two things, glucose and water, right? That makes solution, glucose in water. So this is solution 1, okay? And in this side you have solution 2, that is also glucose solution, but the concentration is higher. You have more glucose here, that's why concentration is higher. So again, glucose and water. Simple diffusion is the movement of the solute. Glucose is solute here, and who is the solvent? Water.
solvent and solute. So glucose is the solute and water is always solvent. So in this condition, glucose will move from high to low. This is high concentration, right? This is low concentration. So glucose will move from high to low concentration, and that movement is called simple diffusion. So now I can say that movement of solute from high concentration to low concentration is simple diffusion. Make sense? Now, water, at the same time, water will move from low to high in opposite direction, and that is called osmosis. So this one is simple diffusion, and this is osmosis. Movement of solvent. So solute moves from what? High to low, right? And solvent moves from low to high, opposite. Okay. And when the solutes move, that is simple diffusion. When solvent moves, that is osmosis. Make sense? Now just think, very simple. What's the purpose? Why they are moving? Why they are moving? To bring same concentration in both sides, equilibrium, right? So if you know the purpose, why they are moving, you can tell which one should move from where to where. Make sense? In this side, which is low? Glucose, right? So glucose should move from here to here. In this side, which is low? Water. So water should move from here to here, to bring equilibrium, right? So it's very simple. Now, facilitated diffusion. Sometimes the chemicals move with the help of other molecules, not directly, with the help of other molecules. That is facilitated diffusion. Sometimes other molecules can carry, for example, you have another molecule here or in the cell membrane here. What will happen? This glucose, you see, the glucose will come and get attached to this and will bind with this, and this guy will then take it to the other side. Okay? So you are sitting on the shoulder of another person and that person is carrying you. You are not directly going, right? So carrier mediated diffusion. Channel mediated diffusion. You have seen when we talked about the integral protein, I have shown you that there are channels through the integral protein. Some channels stay open all the time, some channels stay closed, need ATP to open, but both are channels. So channel mediated diffusion. Okay. So if you are sitting on someone's shoulder and that person is carrying you, that is carrier mediated. If there is channel through the protein stuck here and the molecules pass through that, that is channel mediated facilitated diffusion. It is like that, you know, if you are thinking to cross a lake, water, okay? If the current is in your favor, and you know how to swim, you just can float your body on water, right? And that flow will take you to the other side. Eventually you will go to the other side, right? If you don't know how to swim, then that will not work. You will be drowned. So in that case, you will look for what? Something to sit on it, right? That will take you to the other side, like a boat or something that
you can sit on and will take it. That is carrier mediated diffusion, right? Now, if you want to cross the lake but current is against you, okay, in that case you have to swim, or right, otherwise you will not be able to go to the other side. You have to use what? Energy, swim, or you have to sit on a boat that has what? Engine in it, right? Use energy, otherwise we won't be able, and that is the active process, right? Active process against the gradient, and you need utilization of energy to do that. Okay, in the lab you can simply test simple diffusion and osmosis. Here you see U-shaped test tube, and you have placed a membrane here, placed a membrane, and in one side you have high concentration solution, sugar solution, low concentration sugar solution, and here in this membrane the holes are pretty big, larger than the solutes. So in this case what will happen, the solutes will move from high to low. That is what? Solutes move from high to low? Simple diffusion, very good. And water or solvent will move from low to high, right? That is osmosis. So both will occur. Now, if you put a membrane that has tiny holes in it, and those holes are smaller than the solutes, in this case simple diffusion will not occur. Make sense? Because the holes are smaller than the solute molecules, solutes will not be able to move. So what will happen? Only osmosis. Water will move from left to right, and you can see that because of the water movement or osmosis, the volume of solution in right side will increase, because only water is moving from one side to another. Osmosis only has occurred here. The purpose is bringing same concentration. That is important, you see. Although the volume is high here, low here, but the concentration is same. Okay? That is the goal. How a living cell responds to different types of solution. If you take a solution and put a living cell, for example your red blood cell here in this solution, if the concentration of this solution, for example, is 5 milliosmol, that's the unit of osmolarity or concentration, 5 milliosmol, and inside the red blood cell, in cytoplasm, the concentration is also 5 milliosmol, same, right? Inside and outside, make sense? Same concentration. In that case, this solution is called isotonic. Make sense? Iso means what? Same. Iso means same. Same concentration, right? So, compared to the cell, this solution is isotonic. In this case, no osmosis will occur. Make sense? Because same concentration. So no movement of solute or solvent will occur. Is it clear? Because already equilibrium is there. No movement. So the cell will not change. It will remain as it is. No change in isotonic. Now you tell me, if you take the solution that has concentration 10 milliosmol
this solution is now hypo or hyper compared to this? Hyper. Hyper means more, right? All of you know that, right? Hyper means more. So this solution is what? Hypertonic compared to this, right? So now this is hypertonic solution. So now you tell me which way the water will move. Water always moves from what? In osmosis? Low to high, right? Always moves from low to high. So which way the water will move? Out. Make sense? So water will come out from the cell. Is it clear? Osmosis. So the cell will get bigger or smaller? Smaller. Shrink. The cell will shrink, will get smaller, squeeze. Now if I change this solution, and now the concentration is 2 milliosmol. So now this is what? Everybody? Hypo. Because 2 is less than 5, right? So the solution is hypotonic. So which way the water will move? Always same rule, low to high. So in this case, water will get in. Make sense? From low to high. So what will happen to the cell? Expand, will get big, right? Swelling, or eventually it can burst, rupture. So why you need to know? You need to know this because sometime you take saline inside the body, right, from outside. So you need to know what kind of solution you have to push in. Isotonic, right? So it will not bother the cell. If you push isotonic, it will not bother the cells. If you put hyper or hypo, that may change the shape of the cell, right? Red blood cells in your blood. So it is safe that you use isotonic solution to put in the body. Okay, so let's talk